This is our first unit, flipped classroom, and I want to start off by showing you where you can find the material. It's in Google Classroom under Introduction to Biology. And you are responsible for the slides. There's a total of 39, approximately 43 slides. The rest is just information that I used when I was teaching, but essentially 43 slides you're responsible for, and this flipped classroom is to go into these slides in a little bit more detail and to also help you to catch up if you were absent. So I want to turn the attention to our notebook that we've been keeping, and I asked you in the beginning of the year to come in with pens that had multiple colors. So I wanted to try taking notes with a color coding key where it says black is equal to the main notes, definitions, experimental setup, student answers. Red would mean key terms and vocabulary and examples. Blue equals data, numbers, which we call quantitative. Green is conclusions and analysis, or what we can infer. Purple means connections to the test, higher level thinking, what this means for designing better experiments. So I wanted to take a deeper dive into bias. Bias is a preference for results, and you can find bias on slide number 26. An example is fame, money, or protecting your job. Also on slide number 31. So these are the slides, numbers 26 and 31, that I'm referring to. Now, you would do a blind experiment. This is where, to reduce bias, this is where the subjects don't know if they are the experimental or the control group. Blind experiments reduce bias. Now, a key term is the placebo effect. The placebo effect is eliminated in blind experiments. In experiments with people, sometimes just believing you are getting a treatment can make you feel better, even if the treatment has no real effect, and this is called the placebo effect. Placebo effect equals feeling improvement because you expect the treatment to work, not because the treatment actually works. So that's a key term, placebo effect. Blind experiments eliminate this problem because the subjects don't know whether they are in the real treatment group or the control group. The conclusion is this means any improvement in health is more likely caused by the treatment itself, not just belief or expectations. So here's a scenario. Imagine giving half the class a real headache pill and the other half of the class a sugar pill. If students don't know which pill they got, we can tell whether the medicine really works. So here's some key terms. Blind testing. We're going to do that in red. Without blind testing, students who got the sugar pill might say that their headache feels better just because they thought they got the medicine. In our notes, we were told to draw a picture. When I do a notebook check, make sure that you have this drawn. You might want to pause the video. Figure 1 title is Blind Experiment versus the Placebo Effect. Let's give that a title. And then one person over here and one person over here. This person on the left got the real pill. It's the cure for the headache. This person over here got the sugar pill. Now write a conversation cloud. Both say, I feel better. The conclusion is, this person is better because of medicine. This person is better because of belief. The point of the blind experiment is not to stop the placebo effect. It's to make sure that it happens equally in both groups so that we could see if the treatment group improves more than the placebo group. Or another way to call it is, this would be the control group, and this one would be the experimental group. When I check your notebooks, I want to see this drawing with these labels. Okay, so there is a famous experiment, James Lind, and this comes from a worksheet that I gave you that looks like this, bee and flower experiment, and then we start the James Lind here. So if you were not here for James Lind, or your grade was maybe a 50 because you might have been here for the bee and flower experiment and not the James Lind experiment, and you still need to fill this in, I'm just showing you the paper that I would have given to you and then asked you to watch the reverse classroom to understand. So we're doing the James Lind first, actually, because it's in my notes first, and then I'll get to the bee and the flower experiment. So James Lind experiment took place in 1747. In 1740, Commander George Anson led 8 ships to circumnavigate the globe. Most of his crew died of scurvy. James Lind, a physician, sought to find a treatment and set up the following experiment. He took 12 sailors suffering from scurvy and he divided them into 6 groups of 2. So here's your 6 groups here. The first group that you're going to write in here is cider. And he had 6 groups of 2, so you're going to draw these circles because you're going to be putting in emoji expressions. So group 1 was made up of cider. Group 2 you're going to fill in with sulfuric acid. Group 3 was vinegar. Group 4 was seawater. Group 5 was citrus, specifically 2 oranges and 1 lemon. And group 6 was spices and barley water. So after he gave these supposed remedies, he documented the results. So what you want to do is draw the face emojis. So the group that got cider, it was a partial recovery. So what you want to do is draw, one person got better and the other person did not. Partial recovery. Group 2 that was given sulfuric acid as a treatment, nobody recovered. Both faces should be sad emoji expressions. Group 3, the vinegar group, again, nobody got better, so sad emoji expressions. Group 4, seawater, nobody got better. Group 5, the results were, it was the best recovery. The emoji expressions are all happy faces because there was recovery, full recovery. And then group 6, spices and barley water, there was no recovery. And this is a famous experiment because they realized without vitamin C that's found in the citrus, sailors were getting this disease and thousands of sailors were dying due to a lack of vitamin C. Now, after Dr. James Lind recorded all of his data, came up with his conclusion, he had a peer review. Peer review means that he published a journal with his experimental procedures and the scientific community got back to him and they said, you have a few problems. So for problem 1 here, you're going to write the problem. The first problem was, problem number 1, no constants were defined. So you want to do that in black. What are the constants? What should have been defined in red? Diet, there should have had a defined diet, all sailors. Living conditions should have been defined. Their age, they should have had all been the same age, same severity of illness, and the same amount of water. Because constants are important in experiments. So here's our fix, Dr. Lin. Define these conditions next time to eliminate other variables. So you're going to write all three of those things in here. You're going to write what the problem was, no defined constants, and you're going to do that in black. Tell me the examples of defined constants, and then you're going to tell me what the fix is. And you're going to do this all the way through. Problems 1, 2, 3, and 4. Okay, problem 2. The sample size is too small. There was only 2 sailors per group. The fix is test more sailors because a large sample size equals fewer random errors. Larger sample size equals fewer random errors. Problem number 3. There's no control group. You're missing a baseline for comparison. We need a control group because we need to compare. The fix is add a group with plain water, but tell them it is a supplement to remove the placebo effect. Problem number 4, researcher bias. Dr. Lin's family member was a citrus producer. The fix is you're going to have to use a double-blind setup so Dr. Lind doesn't know either which group had citrus when he evaluates recovery. This is all from slide number 31. Blind, double-blind experiments. Why we do that? We do that to eliminate bias and the placebo effect. And you can read over slide number 31 to understand it a little bit better. All right, now we're going to do the bee and flower experiment. It looks like this. So you should have all 4 of these filled out, this whole side. And now we're going to do the bee and the flower experiment, so let's look at that. You look at a field of flowers and you notice a bee species always lands on the red flowers. You hypothesize the flower color attracts the bees. You run home and you cut out red paper flowers and you place them into the field. I just read this to you. All right, so the first thing that you're going to do is you have to make a line down the paper in the middle, and you have to come up with a title. A proper scientific title would be the effects of X on Y, and we'll make that title on our bar graph when we get to it. But first, let's draw 2 big flowers side by side. Do this flower, and we're going to do this flower. 2 big flowers side by side on the left side, because on this side we're going to do a graph. Above each flower, you're going to draw bees. So you're going to have more bees on this flower, and then just 1 on this flower. This flower is a real red flower, and this one is a paper flower. Okay, now you're going to draw an L here because you're going to do a little graph. You're going to put real flowers, real versus paper flowers, and you're going to write 2, 4, 6, 8. So the independent variable is paper flowers. So paper versus real goes on the x-axis, and your dependent variables are bee visitations. And when you collect the data, you found out on the real flowers, you got 8 bees. But the paper flowers, you had 1.75, statistically speaking, bee visitations. The effects of paper red flowers versus real red flowers on bee visitation. On bee visitations. So again, it's the effects of X. Our X is a comparison of real versus paper. The effects of X, the effects of paper red flowers versus real red flowers on Y, which is bee visitation. So that's how you write a proper title, the effects of X on Y. Now, your observation was that bees preferred landing on red flowers. Your hypothesis was if I make red paper flowers, then they will attract more bees. Your experiment was that you place the red paper flowers in the field with the real flowers, and then you count the amount of bees that show up. And here's our real flower data. Real flowers, 8 bees on Monday, 8 bees on the real paper flowers on Monday, 2 bees only came on the regular paper flowers on Monday. On Tuesday, you counted 12 bees showed up on the real flowers, but only one showed up on the paper flowers. 7 on Wednesday, 4 on the real, 4 on the paper. 5 on Thursday on the real, 0 on the paper. You average that up, and total visitations for real was 8 for the real and 1.75 for the paper. The conclusion is the hypothesis is rejected. Red color alone does not attract bees. The student takeaway: good science equals controlled variables plus large sample sizes plus a control group plus reducing bias. This is the formula for writing strong experiment answers on your test. Now, this is the characteristics of life quiz that we took in your notebook and did it together in class. So if you're missing this, this is the lesson that you're going to want to tune into so that you know what I'm looking for. So the characteristics of life quiz comes from slides numbers 2 and 3. And the way that you remember the characteristics of life is CHORRDM, chord M. C is change, H is homeostasis, O is order, R is reproduction, the other R is response to stimulus, D is growth and development, and M is metabolism or energy processing. So what you want to do here is you want to write a scenario. For example, write this down in black. An insect reacting to you trying to hit it with a fly swatter. So that's the scenario, and then underneath in red, what characteristic of life does that describe? In red, it's response to stimulus. The fly is reacting to you trying to hit it, and the reasoning. This is going to be the definition that you're going to find on slide 2 and 3, so you have to refer to the slide. The insect reacts to a signal in the surrounding environment. It should say environment, to survive. So that's response to stimulus, so that is going to take care of one of the R's in chord M. The next one is shivering when in a snowstorm. What could that be? So in red, the answer is homeostasis, and then the definition. Helps maintain stable body temperature. All right, the third example is a butterfly laying hundreds of eggs. What characteristic of a life is that? That's reproduction. And what's the definition? Reproduction is produces offspring and it's the passing on of genes to the next generation. So with chord M, we did reproduction, response to stimulus, and homeostasis. That leaves change, order, growth and development, and metabolism. So the next thing you need to do, it's your turn. You have to do 1, 2, 3, 4 on your own. Do the other 4 characteristics of life, just like the pattern that I have here. So you're going to think of a scenario for change, for order, for growth and development, for metabolism. You're going to think of a scenario like shivering when in a snowstorm. What is that characteristic of life? And then define it. Again, the answers for the definitions are in slides 2 and 3. But I want you to, when it's your turn, I want you to follow this pattern that I outlined here. And you have to come in and show it to me. I need 7 of them. 7 characteristics of life. I did 3 for you, so you're doing 4 on your own. All right, now we're getting to correlation and causation. Correlation and causation is from a handout that I gave that looks like this. And the back looks like this. And then there's another page, positive correlation, and on the back is no correlation. So correlation means 2 things happen in a way that looks related. They might go up or down together, but one does not always cause the other. For example, when kids wear bigger shoes, they usually read better. Do big shoes make you smarter? No. Older kids have bigger feet and better reading, and that's due to age. So causation is one thing makes the other happen. For example, if you drop an ice cube in the sun, it melts. The sun's heat causes the ice to melt. So here's an easy rule. Correlation means things happen together, but maybe by coincidence. Causation means one thing makes the other happen. So here's correlation on your paper. So what you want to do to understand this concept of correlation, which is things happen together, but one doesn't cause the other, you are going to draw a kid with big shoes holding a book. Then you're going to draw a smaller kid with small shoes holding a smaller book. And then you're going to add a little age arrow showing that it explains both. So it will look like this. Big kid, big book, little kid, little book. You're going to write in blue, big shoes and big book. There's no correlation between big shoes and big. There's a correlation, but big shoes do not cause you to have big books. Little shoes do not cause you to have little books, although there is a correlation. Why would you have big shoes and big books? The causation is age. That's why. So you want to write on the side here, after you draw this, what is the correlation? What is the correlation? And then what is the causation? Correlation is big shoes and big books, or little shoes and little books. But big shoes do not cause big books. What causes this scenario? It's age. So you want to draw an arrow and have age here. In other words, the bigger you are, the bigger the books you can read, and the smaller you are you're not as old, you're smaller, and you can only read smaller books. It's because of age. All right, now we have another scenario. You can turn the page. You have to draw an ice cream cone and a shining sun. You draw an ice cream cone and a shining sun, and you're going to make this correlation. Ice cream cones plus suns happen together. Plus suns, just ice cream cone and the sun happen together. Does the ice cream cones cause the sun to shine brighter? No, it doesn't. There's a correlation though. What is the causation? It's summer. So you're going to want to draw an arrow over, and summer is what causes this to happen. So summer is the causation. It's considered the third factor or the lurking variable or a hidden cause that explains why both ice cream and sunshine happen at the same time. Summer brings hot weather, which leads to more sunshine and people eating more ice cream. Sunshine does not cause ice cream. Summer does. And summer is the lurking variable there. Okay, now you're going to do umbrellas and rain boots. There's a correlation. When you see a lot of umbrellas, you may see a lot of rain boots, but umbrellas do not cause rain boots. Causation is the rain. Rain causes people to use umbrellas and wear boots. Okay, now we're going to focus on causation. That's here. Causation is a concept. One thing makes the other happen. Here's a picture that you have to draw. An ice cube melting the sun. So here's my ice cube. It's melting. So draw like a melting type of picture there. And the sun. And then you're going to want to write causation. The sun, and the arrow means causes the ice to melt. Again, the concept is one thing makes the other happen. The sun causes the ice to melt. All right, now you're going to draw a picture plant growing with water. Draw a watering can pouring water on a small plant, and the plant grows. So here's my watering can pouring water onto the plant. And the causation that I want to write here is water from the watering can causes plant growth. Water, you could just write water causes plant growth. Boom. So that takes care of this page. It's light work, but this one is going to be a little bit more involved. All right, now we're going to get into positive correlation, positive and negative correlation. And positive correlation is as one thing goes up, the other goes up, or if one goes down, the other goes down. They move in the same direction. For example, the more hours you study, the higher your grades usually go. Another example, the taller a kid is, the more they usually weigh. So what you're going to want to do is draw a kid holding a pile of books, and then a kid with a report card that has an A on it. You're going to want to draw that here. So you're going to have a data table. This is how you set this up. Data table, your drawing, and then a graph. So here's your kid with a lot of books, and another kid with a report card, and it says an A. The positive correlation is as your study habits increase, draw an arrow going up. Your grades also increase, draw an arrow going up. It's positive correlation because as one goes up, your study habits, the more books you read, the other goes up, your grades go up. So again, positive correlation. As one thing goes up, the other goes up. Now, we can do a fictitious experiment. Here's our data, and on the x-axis, which is your independent variable, you're going to have hours studying 1 to 6. Your dependent variable is the y-axis. That's the dependent variables. What are you measuring? Independent variables was being changed. And dependent variable, what are you measuring? We're measuring test scores. You spend 1 hour studying, you get a 55, follow it. Or you spend up to 6 hours studying, you get a 94. So on your graph, you're going to write the effects of x on y the effects of studying on test scores. Hours studying is X, label your X. Label your Y, test scores. The effects of X on Y is the way that you write a proper title. The effects of studying on test scores. You're going to space this out, hours 1 to 6, and then the test scores from 60 to 100, 60, 70, 80, 90, 100, plot the data. You find for 1 hour, you got a 55. So you're going to find 1 on the X, go up the Y to 55. Find 2 hours on the X, look at your data, you got a 65. And as you can see, the more hours you study, the better you do on a test. That's a positive correlation, and it moves in this direction. Okay, next we're going to do a negative correlation. So this should all be filled out. A negative correlation is as one thing goes up, the other goes down. They move in opposite directions. For example, the more time you spend watching TV, the lower your grades might get. Or another example, the faster a car goes, the less time it takes to get somewhere. So again, what you want to do on your paper is you're going to want to have a data table. And then you're going to drawing, a line, and then you're going to do a graph. So for the drawing, we're going to start there. For the drawing, what you're going to do is draw a TV. You're going to draw hours on a clock. You sitting in front of the TV. I put little fishies, watching the nature show for too long. And as I watch TV for a really long time, it has a negative effect on my grades. My report card. You're going to draw a report card, F, 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 F. Now, you're going to put the data table to actually do the experiment. Your x-axis hours of TV, y-axis test scores. On the X, you're doing your hours 1 to 6. On the Y, you're doing the test scores. 1 hour in front of the TV, you got a 95. As you spend more time in front of the TV, your scores go down to as low as a 70. So what you want to do is take the time now to graph that right here. So you're going to make a graph. Label it. 1, 2, 3, 4, 5, 6. 60, 70, 80, 90, 100. So that it looks like this. Hours of TV, test scores. When I spend 1 hour in front of the TV, I got a 95. When I spend 2 hours in front of the TV, you have to look at your data. 2 hours is a 90. And so on. Plot your data and notice that you have a negative correlation and the line goes like this. Put your title, the effects of time or hours in front of TV on your test scores. This is a negative correlation. So when your time watches TV increases, then your grade point average decreases. Again, a negative correlation, as one goes up, the other goes down. They move in opposite directions. Now we're going to do no correlation, which is on the back. There's no clear connection. One thing does not seem to affect the other at all. For example, the number of pets you have does not affect your math test score. Another example, your shoe size has nothing to do with how many friends you have. So you're going to draw a kid holding a cat and a math test, but there's no arrows connecting them. So here I didn't put the drawing, but just draw a kid holding a cat. And then next to it, you're going to have a math test score that's bad. Your data table, set it up. So you have 0 pets, 1, 2, 3, 4, 5, 6 pets, and you tabulate math scores with the amount of pets. So with 0 pets, you got an 82. Someone had 1 pet, they got a 95. Another person had 2 pets, they got a 70. 3 pets, an 88, and so on. Now, when you set up your graph, you're going to put the number of pets on the X and the math scores on the Y. When you graph it, 0 pets, you got an 82. 1 pet, you got a 95. 2 pets, you got a 70. 3 pets, you got an 88, and such. You'll come out with a graph that looks like this. This is a graph that shows no correlation. No correlation. The dots are going to look like confetti. Okay? In a negative correlation, the dots are going downhill. On a positive correlation, the dots go uphill. Okay, that ends our lecture. Thank you.